Some background information about this paper is that RAS genes are the most commonly mutated oncogenes involved in human cancers. They play key roles in tumor initiation and progression. RAS proteins are small GTPases that regulate diverse cellular processes, including proliferation, differentiation, migration, apoptosis, and senescence. Thus, identifying the targets that block RAS signaling are of importance in order to develop therapies for cancers driven by RAS mutations. In this research, genes that regulate the essential association of KRAS, a gene in the family of RAS, to the plasma membrane, and thus its signal transduction, were screened for by the use of siRNAs, which are small interfering RNA used to interfere with and cause degradation of mRNA. So the hypotheses or the main goals of this research was to identify key molecules which are required for oncogenic KRAS plasma membrane association, and also whether KRAS has any regulatory genes that could be potential targets for therapy. So the key findings given by the results were that data from the siRNA screening for regulatory genes of KRAS show that when treated with PTPN2 siRNA to knock out the PTPN2 gene, which is the protein tyrosine phosphatase non-receptor type 2 gene, the plasma membrane bound KRAS to cytosolic KRAS ratio was increased significantly. This suggested that PTPN2 plays an important role in regulating the plasma membrane association of KRAS. Next, it was found that the siRNAs were then used to reduce the expression of PTPN2 in KRAS-dependent cancer cell lines, and it was found that to suppress the proliferation of these cell lines. This suggested that the PTPN2 is required for KRAS-dependent tumor cell growth. Further analysis showed that the PTPN2 knockdown increased apoptosis levels of KRAS-dependent cancer cells and proved that PTPN2 is required for cell survival signaling of KRAS. PTPN2 knockdown dramatically decreased the phosphorylation levels of MEK and ERK in KRAS-dependent tumor cell lines, demonstrating that PTPN2 is a major regulator for full activation of oncogenic KRAS and its downstream signaling pathway. It was tested whether PTPN2 regulates KRAS signaling through dephosphorylating KRAS. It has been shown that tyrosine phosphorylation of KRAS reduces its signal transduction activity. So the tests performed show that a knockdown in PTPN2 results in an increased phosphorylation of KRAS and thus demonstrates that the PTPN2 regulates tyrosine phosphorylation of KRAS. This graph highlights the effects of PTPN2 knockdown on the proliferation and survival of KRAS-dependent cancer cells. Here on the x-axis, we can see different cell lines which we have transfected with siRNA and their negative controls beside them. On the y-axis, we can see the apoptotic cell percentage. Analysis showed that the percentage of apoptosis in these KRAS-dependent tumor cell lines was significantly higher than that of the negative controls. This effect of PTPN2 knockdown is no less than that of the KRAS knockdown in KRAS-dependent tumor cell lines. This suggested that PTPN2 is required for the cell survival signaling of KRAS. These images of fluorescently labeled KRAS further provides evidence that the PTPN2 gene regulates KRAS membrane association. The negative control pictures on the top panel show cells with fluorescently labeled KRAS on their plasma membranes only, highlighting the normal functionality. However, on the bottom panel, the PTPN2 siRNA, it shows that the KRAS is more dispersed within the cytoplasm rather than the plasma membrane, highlighting the effects of gene knockout of PTPN2 on KRAS association to the plasma membrane. It was found that the tyrosine phosphatase gene PTPN2 was associated with the increased proliferation of the KRAS-dependent cancer cells, and thus the poor prognosis of patients with KRAS mutation-driven cancers. It was determined that PTPN2 is required for KRAS-dependent tumor cell growth and survival. Thus, the author's conclusions are supported, and KRAS has regulatory genes that may serve as targets for therapy of KRAS-dependent cancers. So the importance of this research is that it has been known that 20 to 30% of human cancers, including a high percentage of pancreatic, lung, and colorectal cancers, are driven by mutations in KRAS. It was found that PTPN2 is required for the effective plasma membrane localization of KRAS, and PTPN2 is required for KRAS-dependent cell survival and, to a lesser extent, the proliferation. These results indicate that PTPN2 gene is a significant regulator of KRAS and may potentially serve as a new target for therapy of KRAS-driven cancers. Thus, inhibition of PTPN2 could suppress KRAS cancer, whereas enhancing tumor immunity. Although science have made great progress in the last 3 decades towards understanding the KRAS signaling pathways, drug development in blocking KRAS function has been ineffective. So further research is required to determine safe ways to develop and administer drugs that inhibit PTPN2 genes in vivo.